Hello dear students. In human reproduction chapter, we have done with the structure of testes and the structure of male reproductive system, as well as we have discussed with the structure of female reproductive system, right? And also we have studied the process of spermatogenesis, how the production of sperm takes place in the testes, right? And now let us discuss with the structure of human sperm. So this is the basic outline of the structure of human sperm. First thing is the human sperm is microscopic. What do you mean by microscopic? Means it is having very minute size. Its size is very small. So how we can conclude it? So we all studied that there will be about 200 to 300 millions of sperms will be ejaculated in a single time, right? That means that it is very huge quantity, right? So it means that the size of sperm is very small. Clear? Now let us discuss with the structure of human sperm. It is basically has four parts. Okay, head, neck, middle piece and tail. From here to here it is called as head. This part it is called as a small neck, and from here to here it is called as middle piece, and the last one is elongated tail. Okay, so now let's describe the each part. First one is head. So here this head part, which consists of elongated haploid nucleus. Can you see this blue color one? So it is nucleus and it is having haploid number of chromosomes. So as we know, sperm is a gamete, right? So gamete will be having haploid number of chromosomes. So ಹಾಗಾಗಿ ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಲ್ಲಿ ಎಷ್ಟು ಕ್ರೋಮೋಸೋಮ್ ಇರುತ್ತೆ, haploid number ಇರುತ್ತೆ. That means that 23, right? And the nucleus is somewhat elongated in structure. ಇದು ಹೇಗಿದೆ, oval shape ಅಲ್ಲಿ ಇರುತ್ತೆ, ಕರೆಕ್ಟಾ? So this is about the nucleus present in the head part. Next important part present in the head is acrosome. Okay, how to pronounce acrosome ಅಂತ ಕರಿತೀವಿ. ಈ ಆಕ್ರೋಸೋಮ್ ಅನ್ನುವಂಥದ್ದು ಎಲ್ಲಿರುತ್ತೆ? ಸೊ ಆಂಟೀರಿಯರ್ ಪೋರ್ಷನ್ ಆಫ್ ಹೆಡ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಕ್ಲಿಯರ್? ಕೆನ್ ಯು ಸಿ ದಿಸ್ ಪಿಕ್ಚರ್? ಸೊ ಇದನ್ನ ಆಕ್ರೋಸೋಮ್ ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, ಈ ಆಕ್ರೋಸೋಮ್ ಇದು ಇಂಪಾರ್ಟೆನ್ಸ್ ಏನಂದ್ರೆ ಈ ಆಕ್ರೋಸೋಮ್ ಅಲ್ಲಿ ಕೆಲವೊಂದು ಎನ್ಸೈಮ್ಸ್ ಇರುತ್ತೆ. ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಆಕ್ರೋಸೋಮಲ್ ಎನ್ಸೈಮ್ಸ್ ಆರ್ ಲೈಟಿಕ್ ಎನ್ಸೈಮ್ ಅಂತ ಕರಿತೀವಿ. ಈ ಆಕ್ರೋಸೋಮಲ್ ಎನ್ಸೈಮ್ಸ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುವಾಗ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇಫ್ ದಿಸ್ ಇಸ್ ಇದು ಓವಮ್ ಅಂತಂದರೆ, ಸೊ ಓವಮ್ ಇಸ್ ಕವರ್ಡ್ ಬೈ ಡಿಫ್ರೆಂಟ್ ಲೇಯರ್ಸ್. ವೈಟಲಿನ್ ಮೆಂಬ್ರೇನ್ ಇರ್ಬೋದು, ಕೊರೋನಾ ರೇಡಿಯೇಟ ಅಂತ ಇರ್ಬೋದು. ಸೊ ಓವಮ್ ಇಸ್ ಕವರ್ಡ್ ಬೈ ಡಿಫ್ರೆಂಟ್ ಮೆಂಬ್ರೇನ್ಸ್. ಈ ಮೆಂಬ್ರೇನ್ಸನ್ನು ಬ್ರೇಕ್ ಡೌನ್ ಮಾಡಲಿಕ್ಕೆ ಕೆಲವೊಂದು ಎನ್ಸೈಮ್ಸ್ ಇದು ಅಗತ್ಯ ಇರುತ್ತೆ. ಓಕೆ, ಸೊ ದೀಸ್ ಬ್ರೇಕ್ ಡೌನ್ ಆಫ್ ಮೆಂಬ್ರೇನ್ ಆಫ್ ಓವಮ್, ಇಟ್ ಈಸ್ ಡನ್ ವಿತ್ ದ ಎನ್ಸೈಮ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ acrosome. Clear? So I repeat, the anterior portion is covered by cap-like structure called as acrosome, which is filled with enzymes that helps in fertilization of ovum. Clear with this? And next part is neck. So neck is present between head and middle piece, and it is very small. ಓಕೆ, ಸೊ ನೆಕ್ಕಲ್ಲಿ ಸೆಂಟ್ರಿಯೂಲ್ ಪ್ರೆಸೆಂಟ್ ಆಗಿರುತ್ತೆ. ಎರಡು ಟೈಪ್ ಇದು ಸೆಂಟ್ರಿಯೂಲ್ ಇರುತ್ತೆ, ಪ್ರಾಕ್ಸಿಮಲ್ ಮತ್ತು ಡಿಸ್ಟಲ್ ಸೆಂಟ್ರಿಯೂಲ್. ಸೊ ಹೈಯರ್ ಸ್ಟಡಿ ಮಾಡಿದಾಗ ಇದರ ಬಗ್ಗೆ ಡೀಟೇಲ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡಲಿಕ್ಕಿರುತ್ತೆ. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಇಸ್ ಮಿಡಲ್ ಪೀಸ್. ಈ ಮಿಡಲ್ ಪೀಸಲ್ಲಿ ಬ್ಲೂ ಕಲರ್ ಇದು ಸ್ಟ್ರಕ್ಚರ್ ಇದೆ, ಹೌದಾ? ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಮೈಟೋಕಾಂಡ್ರಿಯಾ. All are familiar with mitochondria, right? So mitochondria is basically called as powerhouse of the cell. ಯಾಕೆ ಕರಿತೀವಿ? Because it provides energy, right? So ಇಲ್ಲಿ energy ಏನಕ್ಕೆ ಬೇಕು? So mitochondria, it is an energy source for swimming. So as we know, the process of insemination, right? Insemination ಅಂದ್ರೇನು? Release of sperm from the male reproductive tract to the female reproductive tract. Correct? So sperm has to move from the vagina to the fallopian tube, right? As we have discussed the female reproductive system. So for that movement, it should require energy. And ಆ ಎನರ್ಜಿಯನ್ನು ಯಾವ್ದು ಪ್ರೊವೈಡ್ ಮಾಡುತ್ತೆ? ಮೈಟೋಕಾಂಡ್ರಿಯಾ ಪ್ರೊವೈಡ್ ಮಾಡುತ್ತೆ. I repeat, so middle piece possess numerous mitochondria which produce energy for the movement of tail. So tail has to move, right? Then only it can move to the fallopian tube of the female reproductive tract, which provides energy for the movement of tail that facilitate sperm motility, which is essential for fertilization. Okay, this is very important. And the last part is tail. Tail is very elongated. It also helps in motility. Motility andre basically it means that movement of sperm. Okay, so this is overall structure of sperm. And the important thing is whole body of sperm, it is covered with plasma membrane. Clear? So we'll repeat the point. The sperm structure has 4 parts: head, neck, middle piece and tail. Head consists of elongated haploid nucleus, and the anterior portion of sperm is covered by cap-like structure which is called as acrosome. How to pronounce? Acrosome anta karitevi. So it is filled with enzymes that helps fertilization of ovum. And the neck is very small. Middle piece consists of numerous mitochondria which produce energy for the movement of tail that facilitate sperm motility, which is essential for fertilization. Clear? So as I mentioned, there will be release of 200 to 300 millions of sperms in a single ejaculation. But out of that, about 60% of sperm should be having normal shape and size, and at least 40% of the sperm, it should show vigorous motility and the movement irbeku. Artha aita? So even though there will be release of a production of 200 to 300 millions of sperm, only one sperm which is very active and which is motile, that fertilizes the ovum or egg. Okay, so you might be having a confusion between semen and sperm. Okay, is it one and the same or is there any differences? Of course, semen and sperm are different. So sperm will be produced inside the seminiferous tubule of the testes, and the produced sperm will move into the accessory ducts, and then later it to go into the accessory glands, right, where they mix up with the secretion of these glands. The secretions are called as seminal plasma. Okay, so seminal plasma is nothing but it is the secretion of the male accessory glands like prostate gland, seminal vesicle and bulbourethral gland or corpus gland. So the secretion anna in total agi navu seminal plasma anta kariteeve. The combination of seminal plasma and sperm is called as semen. Clear? So when you draw any diagram, the diagram should be neat. This applies for all the diagrams, clear? And the labeling should be in the left side. Okay, so ಯಾವುದೇ diagram ಮಾಡುವಾಗ label ಇರಬೇಕು ಅದರಲ್ಲಿ. Clear? If the question they may ask for 2 marks, then 4 label is must. If the question is for 4 marks, then 8 label is must. Okay, each label carries half marks. And the length of arrow mark, it should be equal. ಅದು ನೀಟಾಗಿ ಕಾಣಿಸುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ? And next thing is you need to write the name of the diagram and the down side. Diagram in the down ಅಲ್ಲಿ ಯಾವತ್ತು ನೇಮ್ ಬರಿಬೇಕು. Clear? So this is about the diagrammatic view. So our next topic is oogenesis. Okay, so So in the previous video, we have discussed with the process of spermatogenesis in detail, right? Hope you remember this. So if you're clear about this process, spermatogenesis, then oogenesis will be very easier for you. Okay, why? Because they are quite similar, but there are certain differences that we'll discuss later. So first, I'll repeat with the spermatogenesis process. So this spermatogenesis process, it starts at puberty. Okay, at the age approximately 12 to 14 year ಇರುವಾಗ ಈ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಅನ್ನುವಂಥದ್ದು ಮೇಲ್ ಸೆಲ್ಲಿ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಇಟ್ ಡಿಪೆಂಡ್ಸ್ ಆನ್ ದ ಹಾರ್ಮೋನ್ ಲೆವೆಲ್, ದಟ್ ಈಸ್ gonadotropin releasing hormone. Okay, so it starts with male germ cell. It is called as spermatogonia and it is diploid in nature, right? And this spermatogonia, which undergoes mitotic division to form primary spermatocyte, and this is also diploid in nature. Why? Because mitosis is equational division, right? So this primary spermatocyte, now it undergoes meiosis first. Since meiosis first is a reductional division, it forms into haploid secondary spermatocyte. Clear? Here it is diploid, here it is haploid. So here what happens? Reduction occurred in the chromosome number, right? So this secondary spermatocyte now undergoes meiosis second to form spermatids. Here the number remains same. Why? Because meiosis second is equational division, it is not reductional division. So this spermatids are not actual sperms. It should get transformed into spermatozoa or sperm by a process called as spermiogenesis. Clear? And the formed spermatozoa or sperms, when they release out from the seminiferous tubule, it is called as spermiation. So this is all about spermatogenesis. Okay, clear? So now let us discuss with the oogenesis. So first we'll start with the definition. So here genesis refers to formation, and simply here oo means ovum or egg. Okay, so oogenesis is nothing but it is the formation of ovum or egg. So it is the process of formation of a mature female gamete, that is ovum or egg. Clear? So next point is it is initiated during embryonic development. This is important. So in case of spermatogenesis, as I mentioned, it starts at puberty, right? But here it initiated during embryonic development itself. Okay, and it starts with the gamete mother cell called as oogonia. ಇದು ಯಾವ ಸೆಲ್ಲಿಂದ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ? oogonia ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. There it was spermatogonia. So here we got two differences. One is spermatogenesis starts at puberty. Here it is during embryonic development, and it starts with the gamete mother cell called as oogonia. There it was spermatogonia. Clear? Now here what happens is, if you consider this as mother womb, ಭ್ರೂಣ ಅಂತ ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ರೆ, if you consider this as female baby developing inside womb. Okay, this is just an example. ಈ ಎಂಬ್ರಿಯೋ ಫಿಮೇಲ್ ಬೇಬಿ ಡೆವಲಪ್ ಆಗ್ತಾ ಇದ್ದಾಗೆ ಇಲ್ಲಿ oogenesis ಆಲ್ರೆಡಿ ಶುರು ಆಗುತ್ತೆ, ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ oogonia ಯಾವಾಗ ಫಾರ್ಮ್ ಆಗಿದೆ ಅಂದರೆ, ಮದರ್ ಪ್ರೆಗ್ನೆಂಟ್ ಆಗಿದ್ದಾಗ ಅವ್ರ ಬಾಡಿಯಲ್ಲಿ ನಾವಿದ್ದಾಗ ಅಲ್ಲಿ oogonia ಅವ್ರ oogenesis ಪ್ರೊಸೆಸ್ ಸ್ಟಾರ್ಟ್ ಆಗಿರುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ ದಿಸ್ ಇಸ್ ವೆರಿ ಇಂಟ್ರೆಸ್ಟಿಂಗ್, ರೈಟ್? So here what happens is, inside the female body, each ovary which produces nearly 1 million of oogonia itself. ಅಂದರೆ ಈ ಎಂಬ್ರಿಯೋನಿಕ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ತಾ ಇದ್ದಾಗಲೇ ಅಲ್ಲಿ 1 ಮಿಲಿಯನ್ oogonia ಫಾರ್ಮ್ ಆಗಿರುತ್ತೆ. ಆ್ಯಂಡ್ ನೋ ಮೋರ್ oogonia ವಿಲ್ ಬಿ ಫಾರ್ಮ್ಡ್ ಆಫ್ಟರ್ ಬರ್ತ್ ಆರ್ ಡ್ಯೂರಿಂಗ್ ಡೆವಲಪ್ಮೆಂಟ್. ಬರ್ತ್ ಆದ ನಂತರ ಅಲ್ಲಿ ಯಾವುದೇ ರೀತಿ oogonia ಆ್ಯಡಪ್ ಆಗೋದಿಲ್ಲ. ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ ಆಗ್ತಾ ಇದ್ದಾಗಲೇ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, 1 ಮಿಲಿಯನ್ oogonia ಫಾರ್ಮ್ ಆಗಿರುತ್ತೆ. ಬಟ್ ದ ಥಿಂಗ್ ಇಸ್, at the end, millions of ovum will not form. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ ovum will be formed only in hundreds, 400 ಅಷ್ಟೇ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ಸೊ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಟು ದಿಸ್ 1 ಮಿಲಿಯನ್ oogonia? ಇದು ಡೆವಲಪ್ ಆಗ್ತಾ ಹೋದಾಗೆ ಡಿಜನರೇಟ್ ಆಗ್ತಾ ಬರುತ್ತೆ. ಕೆಲವೊಂದು oogonia ಅಷ್ಟು ಆಕ್ಟಿವ್ ಇರಲ್ಲ, ಫಂಕ್ಷನಲ್ ಇರಲ್ಲ, ಅಷ್ಟು ನ್ಯೂಟ್ರಿಷನ್ ಅದಕ್ಕೆ ಸಿಗಲ್ಲ. ಸೊ ಹಾಗಾಗಿ ಅದು ಡಿಜನರೇಷನ್ ಆಗ್ತಾ ಬರುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ? ದಿಸ್ ಇಸ್ third thing. And one point clearly ಗೊತ್ತಿರಬೇಕು, during one ejaculation in males, as I mentioned, 200 to 300 millions of sperm will be released. ಹೌದಾ? ಬಟ್ ಇಲ್ಲಿ ಫೀಮೇಲ್ ಬಾಡಿಯಲ್ಲಿ ಮಂತ್ಲಿ 1 ovum or egg will be released per month. ಎಷ್ಟು ಡಿಫ್ರೆನ್ಸ್ ಇದೆ, ಹೌದಾ? ಸೊ ಇವಾಗ ವಿಲ್ ಸ್ಟಡಿ ದ oogenesis ಇನ್ ಡೀಟೇಲ್. ಹೇಗೆ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಸ್ಕೆಮ್ಯಾಟಿಕ್ ರೆಪ್ರೆಸೆಂಟೇಷನಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ, ಹಾಗೆ oogenesis ಅನ್ನು ಸ್ಟಡಿ ಮಾಡುವ. As I mentioned, oogenesis, it starts with the female mother cell called as oogonia, okay, which is diploid in nature. So this oogonia which undergoes mitotic division. Same here also, spermatogonia which undergoes mitotic division to form primary spermatocyte, right? So here it forms primary oocyte. Since it is ovum, so it forms into primary oocyte. Correct? So ಇಲ್ಲಿ ಏನಾಗಿತ್ತು, primary spermatocyte which undergoes meiosis first, ಅಲ್ವಾ? ಇಲ್ಲಿ ಕೂಡ ಹಾಗೆ primary oocyte which undergoes meiosis first. But here what happens is a twist takes place. ಏನಂದ್ರೆ here meiosis 1, as we have discussed, it is divided into 4 stages, right? Prophase first, metaphase first, anaphase first and telophase first. ಇದು ನಾವು ಫಸ್ಟ್ PUC ಅಲ್ಲಿ ಕಲ್ತಿದ್ದೀವಿ. ಸೊ in that prophase first ಅಲ್ಲಿ ಈ meiosis they get temporarily arrested. ಇಲ್ಲಿ ಡಿವಿಷನ್ ಏನಾಗುತ್ತೆ, ಇನ್ಹಿಬಿಟ್ ಆಗುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ? I repeat, oogenesis will start with female mother cell that is called as oogonia, that is diploid in nature, and it undergoes mitosis. Since mitosis is equational division, the formed primary oocyte is also diploid in nature, and this primary oocyte which undergoes meiosis, but here it will get arrested in prophase first of the meiosis. Okay, so ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಫ್ರಮ್ ಬರ್ತ್ ಟು ಪ್ಯುಬರ್ಟಿ, ಅಂದರೆ ಫೀಮೇಲ್ ಅಫ್ಕೋರ್ಸ್ ಒಂದು 13-14 ಇಯರ್ ಆಗೋ ತನಕ ಇಲ್ಲಿ ಯಾವುದೇ ರೀತಿ ಡಿವಿಷನ್ ಆಗೋದಿಲ್ಲ. ಓಕೆ, ದ ಪ್ರೈಮರಿ oocyte will not undergoes any division further till puberty. Okay, but here what happens during that time? During that time, the primary oocyte will get surrounded by layer of cell which is called as granulosa cells. I repeat, primary oocyte, they get surrounded by layer of cells called as granulosa cells. Now it is called as primary follicle. ಇದನ್ನೇನಂತ ಕರಿತೇವೆ? ಪ್ರೈಮರಿ ಫಾಲಿಕಲ್ ಅಂತ ಕರಿತೀವಿ. ಪ್ರೈಮರಿ oocyte ಹಾಗೆ ಇದೆ, ಅದರ ಡಿವಿಷನ್ ಏನು ಆಗುದಿಲ್ಲ. ಸೊ ದಿಸ್ ಪ್ರೈಮರಿ ಫಾಲಿಕಲ್ further they surrounded by more granulosa cells. Now it is called as ಸೆಕೆಂಡರಿ ಫಾಲಿಕಲ್. ಇವಾಗ ಲೇಯರ್ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಆದ ಕಾರಣ ಸೈಜ್ ಇನ್ಕ್ರೀಸ್ ಆಗಿರುತ್ತೆ ಅಷ್ಟೇ. ಓಕೆ, ಆ್ಯಂಡ್ ಯು ಕ್ಯಾನ್ ಸಿ ಹಿಯರ್ ಥೀಕಾ ಲೇಯರ್. ಥೀಕಾ ಇನ್ ದ ಸೆನ್ಸ್ ಥಿಕ್ ಲೇಯರ್ ನಿಮಗೆ ಇಲ್ಲಿ ಐಡೆಂಟಿಫೈ ಮಾಡಲಿಕ್ಕಾಗುತ್ತೆ. ವಿ ಕ್ಯಾನ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ದಮ್ ಆಸ್ ಥೀಕಾ ಇಂಟರ್ನಾ, ದಟ್ ಈಸ್ ಇನ್ನರ್ ಲೇಯರ್, ಆ್ಯಂಡ್ ಥೀಕಾ ಎಕ್ಸ್ಟರ್ನಾ, ದಟ್ ಈಸ್ ಔಟರ್ ಲೇಯರ್. ಕ್ಲಿಯರ್? So again what happens to the secondary follicle is it further surrounded by more and more granulosa cells, and now it is called as tertiary follicle. Okay, ಈ tertiary follicle ಇದು characteristic feature ಇರುತ್ತೆ. So tertiary follicle form ಆಗುವಾಗ you can see there the cavity filled cell, ಹೌದಾ, which is called as antrum. So antrum is the characteristic feature of tertiary follicle. Clear? So ಇಷ್ಟು ಡಿವಿಷನ್ ಆಗುವಾಗ ಪ್ರೈಮರಿ oocyte ಏನಾಗುವುದಿಲ್ಲ, ಅಲ್ಲಿ ಡಿವಿಷನ್ ಆಗುವುದಿಲ್ಲ. ಬಟ್ ಓನ್ಲಿ ದ ಥಿಂಗ್ ಇಸ್ ಸರ್ಟನ್ ಲೇಯರ್ಸ್ ಆಫ್ ಸೆಲ್ಸ್ ವಿಲ್ ಬಿ ಸರೌಂಡೆಡ್ ಬೈ ದಿಸ್ primary oocyte. Clear? So when a female she attains maturity or she attains puberty, that time the arrested meiosis 1, which was arrested in prophase first, that gets completed in her puberty. Okay, so puberty time alli meiosis first complete agutte, which results in two unequal cells. One is very large cell. Okay, so when is very large cell, ಇವಾಗ ಈ ಪ್ರೈಮರಿ oocyte ಇದ್ದದ್ದು ಡಿವಿಷನ್ ಆದ ಕಾರಣ ಇದನ್ನ ಇವಾಗ ನಾವು ಸೆಕೆಂಡರಿ oocyte ಅಂತ ಕರಿತೀವಿ. ಅರ್ಥಾಯ್ತಾ? ಸಿನ್ಸ್ ಪ್ಯೂಬರ್ಟಿಯಲ್ಲಿ ಡಿವಿಷನ್ ಆದ ಕಾರಣ. and it forms a smaller cell called as polar body. Okay, so why the size of secondary oocyte is more? Because it has got certain nutrition which is very essential during fertilization. So ivaga meiosis first aitu. Next what happens, meiosis second takes place, and from the secondary oocyte ovum will be released out, and there is formation of second polar body. Okay, so this is the general idea how oogenesis occurs. But again I'll repeat with the schematic representation like that, like this. That kind it will be very clear for you. Clear? So idu ಒಂದು general ನಮಗೆ idea ಸಿಗುತ್ತೆ. I'll repeat with this. Later we'll study with the schematic representation of oogenesis. So first ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, it will be oogonia, since it is female mother cell and it is diploid in nature, which undergoes mitosis to form primary oocyte, and it is also diploid in nature. And this primary oocyte, they get arrested in which stage? Prophase first of meiosis first. Okay, meiosis first ಅಲ್ಲಿ arrest ಆಗುತ್ತೆ, and there will be no division of primary oocyte till puberty. ಇದು ಇವಾಗ ಎಲ್ಲಿ ಆಗಿರುವಂಥದ್ದು, ಎಂಬ್ರಿಯೋದಲ್ಲಿ ಆಗಿರುವಂಥದ್ದು, ಕರೆಕ್ಟಾ? ಆಮೇಲೆ ಬರ್ತ್ ಆಗುತ್ತೆ ಫಿಮೇಲ್ ಬೇಬಿದು. ಟಿಲ್ ಪ್ಯೂಬರ್ಟಿ ಈ ಪ್ರೈಮರಿ oocyte ಡಿವಿಷನ್ ಆಗೋದಿಲ್ಲ. ಬಟ್ ಫ್ರಮ್ ದ ಬರ್ತ್ ಟು ದ ಪ್ಯೂಬರ್ಟಿ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದರೆ primary oocyte, they get surrounded by layer of cell which is called as granulosa cells, and it forms primary follicle. So this primary follicle further surrounded by more and more granulosa cell, now it is called as secondary follicle. It's a characteristic feature, so there will be theca layer, means you can differentiate theca interna and theca externa. So this secondary follicle is further surrounded by more granulosa cells, which is called as tertiary follicle, and the characteristic feature of this tertiary follicle is formation of a fluid filled cavity called as andron. So by now what happens, meiosis 1 gets completed in puberty and it forms two unequal division, ಒಂದು large cell ಇರುತ್ತೆ ಒಂದು small cell ಇರುತ್ತೆ, but here it was equal division. ಇದು ಕೂಡ ಒಂದು difference spermatogenesis ಮತ್ತೆ oogenesis ಗೆ ಇರುವಂತದ್ದು. There it was equal cell, here it is unequal cell. So the larger cell is called as secondary oocyte and the smaller cell is called as polar body. Later this polar body gets degenerated. Okay, now what about the chromosome here? Is it diploid or haploid? So since meiosis 1 occurred, it is a reductional division, so the chromosome present here it is haploid. Here, so now secondary oocyte which undergoes meiosis second to form again unequal cell. The larger one is called as ovum and the smaller one is called as polar body. Later this will get degenerated, and since meiosis second is a equational division, so we are having here haploid what? Female gamete, that is ovum or egg. And ಇಲ್ಲಿ spermiogenesis ರೀತಿ ಯಾವುದೇ ರೀತಿ division ಇರಲ್ಲ. Okay, that was the general idea of oogenesis. But the thing is, what happens to these follicles, right? So as I mentioned, the tertiary follicle, they matures to form Graafian follicle. Okay, so I repeat, oogonia irutte, oogonia undergoes mitosis to form primary oocyte, where primary oocyte gets arrested in prophase first, right? Later this primary oocyte gets surrounded by layer of granulosa cells, it forms primary follicle, it then develops into secondary follicle and tertiary follicle. By now meiosis 1 gets completed to form two unequal cell, one is larger, right? One is larger, which is called as secondary oocyte, and one is smaller, which is called as polar body. Okay, so this is secondary oocyte. So where primary oocyte gets converted into secondary oocyte, at the same time the follicular cells, tertiary follicle iddadénagutte andre, they get fully matured. By now they will be fully matured, and fully matured follicle is called as Graafian follicle. Okay, ಇದನ್ನ ಏನಂತ ಕರಿತೀವಿ? Graafian follicle ಅಂತ ಕರಿತೀವಿ. Clear? Now what happens is, the secondary oocyte itself, they get surrounded by a layer which is called as zona pellucida. Okay, ಇದೆಲ್ಲ simultaneous ಆಗಿ ಆಗುವಂಥದ್ದು. Zona pellucida, it is a protective layer. By now it is clear that ಅಲ್ಲಿ secondary oocyte ಇದ್ದದ್ದು ಓವಮ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಯಿತು ಅಂತ ಅರ್ಥ. ಯಾವಾಗ zona pellucida ಫಾರ್ಮ್ ಆಗುತ್ತೋ, that means that meiosis second is completed and now it is converted into ovum. Clear? And this ovum will release from the Graafian follicle, and the process is called as ovulation. Generally what happens is, if you consider this as Graafian follicle, ಅದರ ಒಳಗಡೆ ಏನಿರುತ್ತೆ, ಸೆಕೆಂಡರಿ oocyte ಇದ್ದದ್ದು ಏನಾಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ, ಓವಮ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಹೌದಾ? ಡ್ಯೂ ಟು ಸರ್ಟಿನ್ ಹಾರ್ಮೋನ್ಸ್ ಈ Graafian follicle ಏನಾಗುತ್ತೆ, ದೇ ರಪ್ಚರ್ಸ್. ಓಕೆ, ಈ Graafian follicle ರಪ್ಚರ್ ಆಗಿ ಒಳಗಡೆದು ಓವಮ್ ರಿಲೀಸ್ ಆಗುತ್ತೆ. ಸೊ ದಿಸ್ ರಿಲೀಸ್ ಆಫ್ ಓವಮ್ ಫ್ರಮ್ ದ Graafian follicle is called as ovulation. Clear? So this is how ovum is formed. So now let's discuss with the schematic representation. This is the schematic representation of oogenesis. This may ask for 5 marks. So, female reproductive system which produces oogonia in the embryonic development itself, and it is diploid in number, that is chromosome number 46. So, this oogonia which undergoes mitosis to form primary oocyte which is also diploid in nature. Why? Because mitosis is equational division, and this occurs in the fetal life itself. Okay, and this the division of primary oocyte, they get arrested in prophase 1 of meiosis first, and from birth to puberty, there is no division of primary oocyte, but here formation of primary follicle, secondary follicle, and tertiary follicle takes place. So when the female should attain puberty, the division continues. It completes first meiosis to form two unequal cells. One is secondary oocyte, which is haploid in nature. Why? Because meiosis 1 is reductional division, and there is formation of first polar body, and later this will degenerate. This secondary oocyte, this again undergoes meiosis second to form two unequal cells. The larger one is called as ovum, which is haploid in nature. Why? Because meiosis second is equational division. Okay, and this ovum consists of 23 chromosome number, and the formed smaller cell is called as secondary polar body. And later this also gets degenerate, and this is what our female gamete as, which is haploid in nature, and this ovum will be released from the Graafian follicle, and the process is called as ovulation. So today we have discussed with the first one, we have the study module B. We discussed the process of oogenesis and structure of sperm, right? Uh, in the next video, we'll start with the new topic, that is menstrual cycle. Hope you understood the video. Thank you.